So we're going to look at osmosis now, Jack, okay? Osmosis is a fancy way to talk about the movement of water. That's all it is, okay? So try not to overthink it and think it's much more complicated than it is. It's not. It's about water, and it's about moving water from one area to another. That's it. So just like before, there's all your specification. Now, do you see this here, Jack? See the way that you have a practical here? That there practical is... It's not a prescribed, but it can still come up for your practical paper. So you know your theory paper that you have to do? That could potentially be asked as well, okay? Now, these ones here also sometimes comes up, okay? This one here, this one is a prescribed one. So that one there is likely to come up, okay? So when you think about your cells, all right, we have mentioned, so from your year 11, you learn the parts of your cells. Can you think about any of the parts? What ones do you remember? Think about an animal cell. What parts has an animal cell?
Cell membrane, a nucleus, and a cytoplasm.
Brilliant. What about an animal? Or that's what did I ask you? Did I ask you plant? I asked you animal.
Cell.
What about plant?
Cell wall, a vacuole, cytoplasm, nucleus, cytoplasm, cell membrane.
Brilliant. Excellent. So when we think of our plant cell, it is a bit ironic to think that it's actually a bit more complicated. Now, I know you're probably not going to go into A-level biology, but if you did go into it, you'll learn these aren't really realistic. There's way more to it than this, okay? Now, when we think about our cell membrane, right, do you see the one on the outside here? I know that kind of looks like an egg. That's a big thing that we're talking about, okay? Now, we described our cell membranes as being selectively permeable, okay? So, plant cells also have a cell wall which makes them which are totally permeable. Now, in order to understand this, you have to make sure you know what that means. So it's like randomly, right? If I put you standing outside in your clothes and it starts lashing, right? Your clothes will take in some water, okay? Now, if I say stand outside butt naked, none of the water from the rain is going to get into your body, okay? Because your skin is not going to let anything pass through, okay? Now, it will, if you, now, as I meant, I don't smoke, okay? But I know people who do smoke, and they've tried to give up cigarettes by using like a patch. You can also get some like medications, and sometimes they'll put it like a patch on you, okay? Now, the reason why it is that their patch will have some medication in it that will get in through your skin. So we describe it as being selectively permeable. It's going to let some things in, but not everything. So it selects who it lets in. It's like when obviously you're old enough and you start going out, when you go to a bar and there's like I can't think of the word, a bouncer on the door. They will select who they let in, okay? While if you think of the word totally permeable, they'll let everybody in. So if you're going to a coffee shop, they're going to let everybody in, okay? So everybody's allowed in. So that's the difference between them. Now, there's this word here that we really have to understand, and it's known as diffusion, okay? Now, if I was, right, so imagine I'm in my room now, okay? So say I'm here, and you're here, okay? And say I spray a load of perfume around me, okay? Piles and piles and piles, okay? Now, all those wee dots represent perfume, okay? And what you can see is here, that's when I have loads of it. So that's like a high concentration of perfume, okay? Now, over where you are, there's no perfume. But what's going to happen is all those perfume particles are going to start to diffuse. They're all going to start traveling over to you. They're going to travel everywhere. Excuse me. So we're going to go from an area that has a high concentration to over here where you have none. And that's what we call a diffusion gradient. Does that make sense? And that's why, do you know when you go in somewhere and you get, it can be a lovely smell or a horrible smell, or if you're absolutely starving and your mum and dad starts cooking and you can start to smell it in your room, okay? It's because in the kitchen is your high area of concentration. Your bedroom is going to have low area of concentration, but it's going to go from high to low, okay? So diffusion is described as the random movement of a substance from where it's in high concentration to where it's in low. So that's just what I've gone through there, if you have your air and your particles. Now, when we think about osmosis is just a special type of diffusion, okay? So of diffusion, spreading, that's all it is, just spreading from high concentration to low. Osmosis is a special one, but it's about water. It's always about water. So when we think of osmosis, it's about the movement of water from where I'm in high concentration to where I'm in low concentration, but it's across a selectively permeable membrane. Now, I know this wording is slightly different here, okay? So see the way it says from a dilute solution to a more concentrated solution. So imagine I give you 2 glasses, right? Exact same glasses, and we go and get my waddy, right? So in this one here, I have this much my waddy, and then in this one here, I have this much my waddy, okay? Now, if I then put in my water up to the same point, right? So this is all water here and here, okay? So there's my water, and there's my water. Now, because I'll call this one number 1 and this one number 2. Now, if I look at number 1 here, do you see the way I have lots of water and I have a small amount, so I have a small amount of juice, okay? That there is going to be described as a dilute solution, okay? Because it has a small amount of whatever else I'm putting in, and this can be your solute. We can call it a solute because it's something I'm dissolving in it. Now, I know it's 2 liquids, so that will be a dilute solution. Dilute means I've got loads and loads and loads of water, okay? While this one here, number 2 here, this one here has little water and it has lots of our solute, so lots of my my waddy, and that then would be described as a concentrated solution. But it's still about water, Jack, okay? So it's still about, yes, we're now talking about other things in the water. So when we talk about our osmosis, we're saying we're going from an area of a dilute solution has loads of water, to an area with low amount of water. So that's a concentrated solution, but it has to go through a semi-permeable membrane, and that's my cell membrane. Does that make sense? But you have to have all that. Now, what it does, I always mean to fix that silly picture, I keep forgetting about it. So when we think about our osmosis in our cells, the movement of your water is going to go into and out of your cells, and it will depend on what's actually around it. Okay, and I know that sounds a bit random. So now you don't have to know about the red blood cells, but it's actually the easiest one to kind of discuss, to be totally honest. So it's like, say if I had a cell here, right? So if I have a cell here, and I have out here, I have lots of H2O, and in here I have only a little bit of H2O. Which direction is it going to go, Jack? Is it going to go from my beaker into my cell, or from my cell into my beaker? So always think osmosis is about going from high concentration to low concentration. So if
would it be from the beaker into the cell?
Absolutely. So I'm always going to go from high concentration to low concentration. Okay, so absolutely. So I'm going to go from there into there. Now, let's change this up because they might try to confuse you a little bit. Okay, now let's say then, but you're totally correct. And I always bring it back to the water, right? So if I have my cell here, and say now I have a salt solution. Okay, so out here I have a very concentrate salt solution. Now what that actually means, Jack, if it's a concentrate salt solution, that means I have lots of salt, low water, okay? And then in here, in my cell, I have a dilute solution. Now, what that means is I have lots of water and I have little salt, okay? Now, which direction will it go this time, Jack?
The opposite.
Absolutely. Perfect. Brilliant. And it only takes, once you get your head around thinking, right, well, where's all my water? Okay, so it's always going to go from where I have loads of water to where I have a little bit of water. And once you've got your head around that, which clearly you have there now, that makes sense. Could you understand that in school? Did that make any sense in school?
No, not overly.
Not try not to make it too hard. Just always think, right? If they talk about this sugar solution, or if they talk about a dilute solution, or if they talk about a salty solution, always think, right, well, who is more water? Always think about the water. If you always go, right, well, if I have a high amount of water, that's where it's going to move, from high to low every single time. And that's the way I always teach it, because I think see, when you start using dilute and concentrate, it gets really bloody confusing. So when we think about the concentration, it's the measure of the amount of a substance, okay? So that's saying, right, well, how much salt have I in? How much sugar have I in? So animal cells, now I'm sorry about that, it's very annoying. So your animal cells is when the water will move into an animal cell. So for example, a red blood cell, and the cell membrane will stretch and the cell gets bigger. Okay, so if we think about this, the shape of your cell. So if we think now, Jack, right, without even looking any further here, right, you've told me how, say this is my animal cell, right, and there's my cell membrane. Now I didn't mean to use yellow at all. Right, so say I have my cell membrane, and we've said that's the main part, right? So our cell membrane is selectively permeable, Jack, okay? So that will choose what it lets in and what it doesn't let in, okay? Now, if I put this cell into lots of water, okay? So if I have lots of O2 and maybe I have a little bit of salt, what direction will it go?
I don't know.
Think about it, right? So if I have my salty solution, okay? So that's where I have, so it's only a little bit salty, right? But I have lots of water here. That must mean this here has a little bit of water. So what direction do you think it's going to go? In, absolutely. Perfect. Right. Now, if we think that's an animal cell, is there anything that's going to stop that cell from bursting?
No.
No, exactly. So if I put that cell into a beaker with loads of water, okay, the cell membrane is going to stretch, and it's going to stretch so much that it's actually going to burst. And that there bursting is known as cell lysis. And lysis just means cell death. Does that make sense?
Yeah.
Yeah? Now, with your plant cell, it's a little bit different, okay? What does the plant cell have on the outside that's a wee bit different, Jack?
A cell wall.
A cell wall. And what's the function of a cell wall?
It like keeps everything in.
Perfect.
And like it controls what goes in and out.
Absolutely. Yeah. So the difference is, and it is a bit weird, right? Your cell wall, you think about a wall being right, it's got to keep everything in and keep everything out. The cell wall lets everything in. It actually doesn't stop anything coming in. It doesn't care who comes in or who comes out. It's more for structure. It's more for strength. So if I had now the same beaker, right, with still I have this salty solution, but it contains lots of water, okay, I'm putting my animal cell in this time, and we know it has that extra part. So now I have a cell wall and I have my cell membrane, okay? Now, what direction is my water going again though, Jack?
Going in.
It's going to go in. Perfect. Now, what about this time? Will this burst this time, Jack?
No.
Brilliant. And why not?
Because like the wall can like let the water out if it's becoming too much.
It's not that it's going to let it out, but it just withholds its shape. It just keeps the shape. And that there, see, keeping that shape. And this is the part that I've blocked out in here with that wee diagram. That's known as turgidity, okay? Now, I'm going to put up the wee part and I'll get you to write in what I want you to take down next. It's just blocked. Now, this is the wee part that I've blocked out there. Now, you know the way when we have our plant cell, Jack, just don't take it down for 1 wee second, right? If that's my plant cell, we know, right, there's my cell wall, which is going to keep its shape, okay? You have your cell membrane, selectively permeable. It allows some things in, not everything. What do you call the big giant thing that looks like a lake?
Vacuole.
Absolutely. So there's my vacuole. So what happens is if I have this in my beaker that has lots of my water, okay, you are 100% correct telling me the right thing. It's going to move in. And once it moves in, Jack, it actually moves into the vacuole. See that there part? So it's going to move into the vacuole. So the vacuole ends up getting bigger. Does that make sense?
Mm-hmm.
Good. And we're going to look at a diagram of that. I'll let you take down that wee part there. That's just the wee part that was cut off on your card. Okay, so the force of this here, right? So see this here pushing out. That's known as our turgidity, okay? So when something is really turgor, it means like it's swollen, okay? And it is quite like, this sounds gross, okay? But you know when you get a burn and you get a blister and you can kind of push it, okay? That sounds disgusting, but that's what turgor is. Your skin has been turgor. So the force of this will increase the turgor pressure within the cell and it makes it quite firm or swollen, okay? Now, the pressure created by the cell wall stops too much entering and prevents cell death, which is our cell lysis. So unlike the animal cell here, that if it keeps taking in water and keeps taking in water and keeps taking in water, it's going to burst because there's nothing there that's going to stop it. With this one here, because I have my cell wall, the cell wall will get to a point and goes, enough's enough, no more. And this here, our vacuole that is here, is going to take in the water and it pushes against it. So it causes our cells to actually get thicker. And what it's going to happen then is once it gets thicker, it'll push against your cell wall. Now, the opposite of that, Jack, say I do the opposite, right? And I put my cell, my plant cell, so there's it there. We know we have our cell wall, our cell membrane, right? And say in this one here, I have lots. So say I have my water in my cell, but I put it into a really concentrate salt solution. Now, break that down what that means, Jack. If I'm in a concentrate salt solution, okay, does that mean I have lots of water or a little bit of water?
A little bit of water.
A little bit of water, absolutely. So I only have a little bit of water and I have lots of salt, okay? Now, because I have a little bit of water there, what direction is my diffusion gradient or my osmotic gradient? So all these I know sounds dead fancy, and it's only a fancy way to say what way is my water going? They all mean the exact same.
Would it be going out?
Absolutely. Perfect. So it's going to go from my cell into my beaker, okay? Now, if you think about what do you think is going to happen to my vacuole then, Jack? So if it's losing the water, right? So the water that was in my vacuole is coming out and going into my beaker. So what do you think would happen then to my vacuole?
It's going to get smaller.
Absolutely. Perfect. And what actually happens is my cell becomes what we call flaccid, okay? They're no longer turgid, which is kind of like swollen. And what is going to occur is my cell wall, my cell membrane actually starts to pull away from my cell, which I'm going to show you in a wee second. So this you've actually covered most of it there now, okay? So our movement of water will depend on the cell and what it's around. Now that's actually very much repeated from the last one. I didn't even realise that. That's my fault. Now, all of this you've done, okay? We've actually done all that there now. I need to kind of change that up. Now, this here is what we've also covered, okay? Now, you have told me about my different solutions and we're going to give them new names as well, okay? So if I have a really concentrate solution, so just like my salt solution, you told me it had a little bit of water. We can also call that a hypertonic solution. Now, Jack, they're not supposed to use these words because they're technically not in your course, but I have seen them come up. So that's why I go through them so you don't panic and go, I don't know what that means, okay? So if I have this hypertonic solution, that means it's really lacking in water. So I have a little bit of water, but I have loads of whatever else, okay? And look what we have. This is what we talked about a wee second ago. So look at my water's going out. Now, don't worry about this here. That's the red blood cell. It's just to show you what it looks like. That would be asked at higher tier, okay? But my water is coming out of my cell. And look, it's been pulled away. And they love to get you to draw that. That's a really common question, is they ask you to draw this one or draw that one, okay? So in this one here, what's happening is the cells have a higher water potential than my solution. So imagine it's in my beaker, okay? We're saying our water's coming out of my cell into the beaker, and that means I'm going to have these like shriveled up cells. Does that make sense, Jack?
Yeah.
Yeah. Now, if I put in what I call an isotonic. Now, isotonic, you've probably heard of that word before, isotonic. I think you can get like Lucozade, Isosport and all this. Iso means it's the same. That's all it actually means. So it means I have, if I was to put this here, right? So say I put this in my beaker, okay? And say my cell had a set amount of water, but my beaker had the exact same amount of water. What do you think would happen with that one? So if they have the exact same...
It would stay the same, basically.
Totally. Absolutely. So it's like some water's going to come out, but some water's going to come in. But it's going to do it in a way that it's going to end up balancing out and it's going to look like nothing's happening. Okay, it's like a busy restaurant. If you have a restaurant, and that's actually on your chemistry course, it's known as dynamic equilibrium. It's like if you have a really busy restaurant, okay, you don't have the same people sitting in the same seats the whole day. You'll have people coming in and people going out, and every seat will be full at all times, not by the same person. And it's the same with this. So some of my water is going to come out and some of my water will stay in, but it'll end up being balanced. And then if I have a hypotonic solution, that's when I've loads of water, but I only have a little bit of salt, a little bit of glucose, a little bit of whatever they want to talk to you about, okay? So what's going to happen here is my water's going to move in. And look at my cell there. Do you see the way it's kind of like swollen, a bit chubby? Look at my vacuole. My vacuole has now been pushed against your cell membrane. And that's what they're looking for there. Does that make sense? But they're the two. If they're going to ask you, they'll ask you to draw one of those two. So this, now you could have to demonstrate this, okay? Now you've obviously done your practical. What was your biology practical on? Did you have osmosis?
Yeah, it was osmosis.
Was it potatoes or what was it? Can you remember?
Yeah, it was something to do with potatoes, like putting them in the solutions.
And to see the length. Yeah, that's this, okay? And that's all it's about now. And hopefully now you'll understand a bit more of what you're actually looking for, okay? So what we have, and again, Jack, this could still come up as your theory. It can come up in your theory or it could come up in the practical paper. So what we do is we cut 5 equal lengths and width of potatoes, and we weigh and dry them, and we place them in different concentration of sucrose for a set time, dry and reweigh. Sucrose is sugar. It's just a type of sugar. So the percentage change in our mass or our length is going to be calculated from our equation. So you have your percentage change will always be your percentage change is your change over your initial. There should be a divide sign there, pet. Times that by 100. Okay. Now, what you'll see here is imagine, let's talk about these. So forget about all the writing for a second and let's see if we can actually work it out. Right, so let's say I have 5 different solutions, okay? I have 5 potatoes that are the exact same. Now, that doesn't mean whenever I put them in, Jack, they're all the exact same mass. There's no way you're going to have 5 potatoes the exact same mass, okay? But it's not going to matter because you're looking for the mass change in that potato. Does that make sense? So it doesn't matter that they're not all the same. Now, say I have in this solution, okay, I just have just water, okay? Now, if I've just in this one here, I'm going to make these numbers up. I have a 1 molar sucrose solution, okay? In this one, I have 0.2 molar sucrose solution, 0.3, 0.4, okay? Now, here's 0.01, okay? Now, what you can see here, what's happening to the amount of sucrose, which is my sugar, as I'm going up? So what's happening to the amount of sugar, Jack?
I think increasing.
It's increasing, absolutely. So in each of my solutions, now we're talking about the solution here, I've increased sugar, okay? Now, if I'm an increase in sugar solution, that means it's becoming more concentrate. Now, bring that back to my water, Jack. What does that mean for my amount of water?
There's less water.
Less water, absolutely. So I'm going to have less water as I'm going up, okay? Now, if I have 5 potatoes in each of them, okay, we're saying as I'm going up, I'm getting less water, okay? Who's going to change the most, Jack?
The one at the very end, 0.4.
The 0.4. So for in this one here, because this one here, so if each of my potatoes has water, okay, and we're saying that this solution here has the most sugar, least water, okay, what direction is my gradient? Am I going to go from my potato into my water or vice versa?
Vice versa.
So we're going from the solution in. Think about that. So if that's my least water, that's the low concentration, which means my potato has to be my high concentration. So
then it's coming on.
It's going to become the other way, absolutely. So we're going to go from here, okay? So then, who would be the one that's going to change the most in terms of mass? Who's going to be the lightest or the heaviest, whichever way you want to look at it?
0.01.
0.01, absolutely. Because this one... No, hold on. Now, I actually have not managed to confuse myself here. No, it'll be the other way around, okay? So this one here is going to lose the most water, okay? Because it has the most sugar. Does that make sense?
Yeah.
And so sometimes, Jack, you have to think about it. Like even now, I'm like, am I right here, okay? So this one here, this potato here, should be the least in mass, okay? So that should lose the most. It should have the greatest change because it's losing the most water. Does that make sense? So it would, this one here, I'll change that there up. I should have the biggest change in it. Does that make sense?
Yeah.
Yeah? So let's look at this. So if I have Now, I've just put them all in sucrose solution, okay? Say I had, right, so let's look at the decrease one first, okay? So if I have a decrease in mass, that meant the potato cylinders were placed in a strong sucrose solution, and they're going to lose our mass because our water will go from high area to a low area. Does that make sense, Jack?
Yeah.
Perfect, right? So that was the one that we looked at. Now let's change it up, okay? So imagine then, right? Say this first one here. Say I put this one here into pure water, okay? So that means here, my solution has only water. It has loads of H2O, okay? What direction is my gradient this time?
It's going out.
Is it going from the potato into the water or from the water into the potato?
From the water in the potato.
Absolutely. And what should happen to the mass of my potato now, or the length, whatever you're measuring?
It should increase.
It should increase. Absolutely perfect. Brilliant. So if my potato cylinder it would increase in pure water or in a weak solution, okay? It depends on what solution we're using. Because we know it'll always go from an area of high concentration. So it depends who is your high concentration. Does that make sense?
Yeah.
And now what about if we had, say this one here, right? Now I know I'm changing these up on you. Say I did my experiment, right? I measured my potato and it was, I don't know, I'm making these numbers up, 1.1 gram, okay, at the start. And at the end, it was also 1.1 gram. So I have no change. What would that tell me?
It's in a neutral solution. It was just an equal consistence.
Brilliant. Excellent. So it means I've used my isotonic solution. It means that my potato and my solution must have the same concentration of water, and try to use that word concentration. Brilliant. Excellent. So we can also demonstrate this using a little experiment known as the Viscon-Tubing experiment, okay? Now, all you do for this one here is you get, and it looks like a piece of plastic, right? It really, really, really does. All you do is you get this Viscon-Tubing, okay? And they might ask, why are you using that? And you're using it because it's to model the cell membrane. Because remember, the cell membrane is what's letting some in, some out, okay? Does the cell wall choose what it lets in? Is it selectively permeable, Jack, or is it fully permeable?
Fully permeable.
Yep,
Yeah,
yep, you're right. Brilliant, okay? And even though, think about the wall's more to do with strength, okay? So with our selectively permeable cell membrane, okay, what you do is you tie a knot. You're going to fill it with water or sucrose solution, whatever you want, okay? You're going to tie another knot. You weigh it, okay? So you dry it off. You have to dry it off so that there's nothing on it. Then you're going to put it into a solution, okay? You tie your knot, put it into your solution. You leave it for about an hour, and then you reweigh it, okay? And you have to dry it off before you reweigh it. And what you'll see is exactly what we like. It's the exact same experiment as the potato. It's just we're using Viscon-Tubing. Does that make sense?
Yeah.
Now, you're going to tell me what's happened in this though, right? So I'm going to take just the exact same experiment here. Now, imagine in this experiment here. So I'm going to call this experiment 1, okay? At the start of my experiment, my Viscon-Tubing weighed 2.4 grams, okay? Now, my solution here, so this is a sugar solution, okay? So glucose solution, sucrose solution, doesn't matter, okay? And in here, I have pure water. I want you to tell me what should happen to my mass in this one here. So think about who has loads of water, who does not have loads of water, what that's going to mean. So start off with the sugar solution. Break it down bit by bit. So tell me about the water content in that sugar solution.
So that solution will go into the... what's the thing called?
Viscon-Tubing. So no, don't worry about that just yet. Tell me about the sugar solution. So does it have high concentration of water?
It's a high concentration of sugar.
Yep. So high concentration of sugar. And what would that mean for my water?
So it's a low concentration of water.
Good. Brilliant. Right. Now that you've figured out what your solutions are, where's your gradient? So who am I going from? And always remember what osmosis is. Osmosis is the movement of your water from an area of high concentration, think of the perfume, to a low concentration. I'm always going water, high to low.
So it's going in, would it be?
So if this one here is pure water, what does that mean for my water concentration in it?
It's more water concentration than sugar.
No. So think about it, right? So if I only have water in here, only have water, so that means I have a high concentration of water and I have nothing. So that's like my low concentration of sugar. So if I have my high concentration there.
So it's going out into the
Brilliant.
sugar solution.
Perfect. So it's going to come out. Great. Right. So now I take my viscous tubing out. I'm going to reweigh it. What should happen to this mass?
Should go down, would it be?
Absolutely. Perfect. It has to decrease. Good. Right. You're going to explain this one to me. Okay. And I know you're probably like, oh, geez, I just did this one. Right. So imagine I have pure water here. Okay. I have sugar solution here. Okay. So think about where's my high, where's my low, where's my gradient. My mass at the start was still 2.4 grams. Okay. I want you to tell me what's going to happen here.
So the pure water is going to go into the sugar solution, and whenever it's taken out, the mass will increase.
Absolutely brilliant. Absolutely. And what do I call that piece of apparatus? What's it called?
Viscous something. I can't remember the one.
Viscous tubing. Good. Why do I use that? What's the point of using that? What is it showing?
The concentration.
No, but what is it? What is it like? Modeling. So what is it pretending to be?
Cell.
Cell what though?
Cell membrane.
Cell membrane. Perfect. That's what it is, Jack. So your viscous tubing is acting like your cell membrane. It's like a piece of plastic that has tiny little holes that's going to allow the water go in and out. Okay. Right, Jack. I actually must add this here in. So, Jack, what I want you to do for a second is I want you to take down, do you see this diagram here? Now, it doesn't have to be a work of art, Jack, okay? So, I want you to take down this. This is coming from your year 11, but you have to understand this to understand transpiration. Okay, so in order to understand transpiration, you have to understand the structure of your leaf, okay? Now, what you have, don't worry about all this here, okay? What you have is at the top here, you have your cuticle, okay? Now, your cuticle is made of wax. It's a waxy cuticle, kind of like a rain jacket. And the whole point of this here is to prevent water loss. Okay, because we don't want, if you think about the top of your leaf, so imagine my hand is a leaf here, the sun's up here. Okay, that's where all your evaporation is going to occur because it's going to be shining down on it. So we don't want that to happen. So we have our waxy cuticle. You have your upper epidermis, okay? Now, do you see the way it's really thin? It's quite thin and it's very structured. There's actually no chloroplasts, okay? So we would describe your upper epidermis has thin and transparent. It will allow all the light to go through because we want it to go through to this area here. Now, do you see this area here? This is known as my palisade mesophyll. And look at the structure of it, Jack. It's really like tightly, densely packed in this really structured column, okay? And it has loads of chloroplasts. So there's where loads of my photosynthesis is going to take place because they're in a column. They're really structured. Column, tightly packed. And it has loads of chloroplasts. And we know your chloroplasts is where your green chemical, your chlorophyll, is produced. Okay, and that's what actually traps the UV light from the sun. Here you have your spongy mesophyll. Now, look at the shape of this, Jack. What do you notice different between our shape here and our shape here?
They're all different shaped compared to the top ones.
Absolutely. And you see all these kind of gaps in between? That's air spaces. And because I have airspace, that's where my diffusion is going to occur. Diffusion is just your spreading of your gases, okay? So that's where the diffusion occurs because I have all these spaces. And that there is known, this area here is known as my spongy mesophyll, okay? And here I have my stoma or my stomata. And your stoma is your stomata, or your stomata is where gas exchange occurs, okay? So it's where I'm going to take in my oxygen, give out my carbon dioxide. Or sorry, other way around. Take in my carbon dioxide, give out my oxygen. And then do you see these two cells here? These are known as guard cells. And they're the ones that control the opening and closing of this here. So it's like the guards. The guards are like the police in Ireland, okay? They control who comes in, who comes out. It's the same as this. They're going to control this stoma. Does all that make sense?
Yeah.
Good. Brilliant, Jack. Now, this is where it gets a little bit harder, okay? I don't like this part either, okay? We have to look at something called transpiration. Now, transpiration is the evaporation of water from your spongy mesophyll cells, followed by the diffusion of water through the air spaces and out of the stomata. Now, I know that sounds ridiculously hard. Now, to understand this properly, Jack, I need to take you back a wee bit and I need to go through the structure of a leaf from year 11. So what we're saying is our transpiration, look what it actually is. It's the evaporation of water, okay? We can make this sound as fancy and as difficult as we wanted to, but that's what it's about. It's about the evaporation of our water. Osmosis was the movement of our water. Our evaporation is specific. So transpiration is this here evaporation, and it's going to go from my spongy mesophyll cells. So that was the cells that had, and I know this diagram is the same diagram. All it is kind of shifted on its side, okay? It's the area that had load of those gaps, followed by the diffusion of the water through the air spaces and out of the stomata, okay? So the stomata was like the opening. So do you see what I mean? So we've gone through the top half, right? So we're saying our evaporation, our water, okay, it can evaporate through here and it can come out through there. Does all that make sense?
Yeah.
So our mesophyll cells are the cells that have our few chloroplasts, and it has a large area because of this that'll allow the diffusion of our gases. So we mentioned perfume earlier, okay? Perfume's actually, it's a vapor, okay? But it's still technically, it's a liquid that has been condensed to a vapor. But when we talk about diffusion, it's the spreading. So if somebody lets off a stink bomb, okay, it's all gas particles, okay, it's still spreading out, and it's the same with this here. So we're saying our mesocells, these ones down here, they had an awful lot less of the chloroplast compared to, remember the palisade cells that were in like the columns, okay, and they had loads of them. Your stomata is the tiny little holes down there, okay, and they control the gas exchange. So most of my water is going to enter a leaf and it will exit the leaf into the atmosphere. So that's what's going to happen, and it creates this continuous stream, and that's known as a transpiration stream. So some goes in, some goes out, kind of like when we breathe, we're taking oxygen, we give out carbon dioxide. It's this continuous stream, okay? Now, why this is actually important is it's going to give the leaf a continuous stream of water, because we know we need water for photosynthesis. It'll continually transport the minerals from the roots to the leaves and to other parts of the plants. It provides plant cells with support from turgor. Okay, now we've looked at turgor. Turgor is like the swelling. Do you remember we said taking in the water? The water will enter my vacuole. It'll push against the wall and it makes it kind of swollen. And it provides water for transpiration. Now, if my water is quite low in availability, what the plant does, very clever, is it stops this water loss. Okay, so it stops the evaporation actually occurring. And how it does this is it takes the stomata here and it closes it. So the guard cells actually shut it up, okay? Because we don't want to lose all our water, okay? Now, what affects our transpiration? And this I find this a bit difficult, okay? There's a few things that can actually affect it. So it's like, because we know, just kind of keep going back to, well, what is transpiration? It's the evaporation of water. So we're saying, right, well, what is going to make my evaporation easier? What is going to make it less? So the surface area, okay, and this is not classed an environmental factor. So if I have really small leaves, they're going to contain less stomata, causing a decrease in transpiration. I'm going to have less evaporation. So when you think of your leaf, okay, so your leaf is like this here, Jack, okay? Now, that's the side that's facing the sun, okay? Clearly not to scale, and that's why I'm not an art teacher, okay? But underneath, the underneath side of my leaf here, that's where my stomata is, all right? So if I have tiny leaves, I'm going to have less, okay? So if I wanted to look and experiment at this here about how I could change it up, right, well, I could remove leaves, and that would allow me to work out, right, well, if I have less leaves, will that have an effect on transpiration? Absolutely. I'm going to lose less water if I have less leaves. I could look at wind speed. So a higher wind speed will actually increase the rate of your evaporation, causing an increase in transpiration. Now, logically think about that, Jack, right? Think about what we said about osmosis. So if I have my leaf here, right, and it has lots of water, okay, and outside here in my atmosphere, I have only a little bit of water, okay, where does the water want to go? Where would my gradient be then?
It would want to go out.
It'll want to come out, absolutely. So it's going to want to come out, okay? It's going to follow the rules. So if I increase my wind speed, that means my wind is moving the water along, or moving the water in the atmosphere along. It's going to be constantly low. Does that make sense? And that would increase our transpiration. And how I would kind of do a mock of this, or a fake one, would be I'd use a fan in an experiment. Temperature. A higher temperature will increase the rate of evaporation. Again, exact same. So if I have a really hot day, that would mean I have only a little bit of water in the atmosphere. My water's going to move out. But it's going to keep coming back to osmosis, Jack. Does that make sense? If you can keep thinking, right, well, where is my water? How I would mock that in an experiment is I would use a heater. Humidity, a higher humidity level will reduce evaporation. Now, what higher humidity means is it means that I have loads of water in the atmosphere. So if I have loads of water here instead, what direction will it want to go, Jack?
Want to go in.
It'll want to go in, absolutely. So if it wants to go in, that means it's going to have the opposite effect for transpiration. It means I've less transpiration. So that means that, because that's evaporation. So it's you're linking your evaporation to your osmosis. If I have light intensity, if I change it, right, darkness will cause the stomata to close, causing a decrease in transpiration. If I have no light, I have no photosynthesis. Okay, if I have no photosynthesis, the plant will try to hold on to the water that it has. Does all that make sense?
Yeah.
Good. So we can also use what's called a potometer. Okay, now a potometer is a piece of apparatus that is designed to measure this water uptake in a leafy shoot and the factors that affect the rate of water uptake by the plant. Now that sounds ridiculous, okay? Basically, we're saying we want to see how much water my plant takes up, okay? So spreading of water, we're looking, it's osmosis again, okay? But we're trying to actually measure it this time. Now, the issue with this here is I cannot provide an accurate value for the amount of water that is being transpired, because some water will be used by the plant to provide things like for photosynthesis and for turgor, okay? So we're basically saying, right, well, if I want to measure how much water I'm losing, I have to do that by measuring how much water is going to be replaced. That's what I'm doing in this. Does that make sense?
Yeah.
And it takes a wee bit of time to get your head around that. Now, I have two types of potometers. I have the bubble potometer and I have the weight potometer. Now, what we have is we take our plant here, okay? And what you're going to do is you're going to use this potometer to measure, you're kind of going backwards. So you're measuring how much water is lost by measuring how much water has taken in to replace what it has just lost. So as water is transpired from the leaves, the shoot absorb the water from the potometer. That is a fancy way, Jack, to say has the water is evaporated from the leaves, the shoot is going to absorb the water from the potometer, okay, because it wants to replace the H2O. Okay, it wants to get that back. So the distance the air bubble moves in a certain period can be used to calculate our rate. Okay, so here's our air bubble. And basically we go, right, well, if I know how much water is here, right? So if I know where my air bubble is, I can work out then how much water has actually been taken up. Now, what they could ask you about this, Jack, is how you set this up. And it's a bit of a complicated setup. This has to be set up underwater to prevent the entry of unwanted air bubbles. I only want one air bubble because I'm using that to measure. Okay, if I have two, sure, how am I going to know which one to measure? We need to make sure the junction between the shoot, so here's my plant here, and the potometer is sealed. And what you do is you use petroleum jelly, that's Vaseline, okay, to stop any leaks actually occurring. The apparatus then has to be reset using a reservoir or a syringe so that the water uptake in the same leafy shoot can be measured, making the investigation more reliable. In science, when you talk about reliability, reliability is repeating, okay? So we need to be able to repeat this experiment to make it reliable. It can also be used to reset if I want to change my environmental factors. So we mentioned different environmental factors we could test, okay? So temperature, humidity, okay? So you could change it up there and to do that, you'd have to reset it. So when you're investigating the effect of an environmental factor, it is really important you keep everything else constant, okay? In any experiment, there's three factors, Jack. You have your independent, and your independent is what you change. And that's one thing. You will have your dependent, and your dependent is what you measure. So for in this example here, you're measuring the distance that air bubble went. And then you have your control. Your control factors is all others, and they have to be kept the same. If you change one of them, you've invalidated your experiment. It's not fair anymore, okay? If we had, so just like what I've already mentioned, okay? Now, you are not going to be asked this in terms of write me a method. They're not going to ask that. So what we do is we just set up the bubble photometer, note the position of my bubble, so where it's starting, leave it for a period of time, because in that period of time, Jack, what's going to happen is my water from my leaves, okay, my H2O in my leaves is going to go into the atmosphere. So then my water is going to be taken up by the roots. And all you're going to do then is going to calculate the rate. Look at the new position of your bubble and calculate the rate. And you just do that a few times, okay? Now, there is another one we could use, is we could use a weight photometer. And this apparatus and the plant is weighed and it's left for a period of time. And then we can calculate our water loss. So all we're doing here is we put our plant, our shoot, into our water, okay, stem down. We know our leaves has our water, and it's gonna evaporate, okay, it's gonna get lost. And what's gonna happen is water will be taken up. And we just look at the difference between our mass calculations. Now, I have a layer of oil here as well, because, and the whole point of having a layer of oil is you don't want water to evaporate off. So you need to make sure that that is kept there the whole time. Does that make sense? Because otherwise it would skew your results. But all you're doing there, Jack, is you're looking to go, right, well, if I can work out how much water I'm taking up, that tells me how much I have lost. Now, your last one is we have this here. It's known as the washing line method, okay? And all you do is you take a number of leaves from a plant, you put Vaseline on them to cover the stalks, and that makes them waterproof because no water will be able to get in. You measure the mass of each leaf. You put them on a string, like a washing line, a piece of string with 2 retort stands, and then you leave it there. You might have, like, to look at water loss. You might have a fan or something. You might be looking at temperature. And then after a period of time, you'll just reweigh them. And from that, you'll get the average or the loss then of each of them. Does that make sense?
Yeah.
Have you any questions? That's that topic done, Jack. Have you any questions on that?
No, not really.
Good. Right, how do you feel about those 2 topics then?
The other topic that we'll have to do is where I struggle more. These 2.
The other one, the chromosomes one. Grant, we'll do it. I'm going to do it on Thursday with you. They were the ones that your mom had said that you found difficult, those ones there. Are you happy enough?
Yeah, I just struggle with, like, the heart, and, like, I know what they're all called in the heart, like different, like, things like the vena cava, but it's a case of, like, where they are and stuff. Yeah, grand. We'll just go back over it, look at a few questions, Jack, and your mom has my number. You can text me, it's no problem at all, or get her to text me and I'll answer any questions. But on Thursday, Thursday, I can't remember what day it was. Thursday we'll do chromosomes. Now that's a really long topic, so I imagine it'll probably take an hour and a half anyway. What other topic do you want to go over if we get on to another one? Let me have a look here.
Variation's very simple. And you have...
What else is in the spec?
You have variation and selection, very simple. So it is that one I wouldn't be too worried about. You also have health and disease. It's quite long. And you have reproduction. 100% you're going to get reproduction. It's always comes up. So maybe did you find the wee cards useful?
Yeah.
Well, I'll send the other 3 sets anyway, and sure you can have them anyway, and sure we can have a look and see. I have it all prepared anyway. Is that okay?
Yeah, that's all right.
No bother, Pet. Here,